Good evening, all of you. Am I audible and visible? If I'm audible and visible, can I have a thumbs up, please, so that we can start the session? Hi, Rishiraj. Are you there? So, if you joined the session for the first time, this is Dr. Shanmugapriya. I'm the Unacademy educator for biochemistry. Good evening, Ankit Sharma. Good evening, Vinod Jadhav. Yes. So today's session is all about biochemistry in 2 hours. So I will try to cover as many MCQs as possible in biochemistry, and this discussion will be based on previous year MCQs. I've chosen few questions in which were asked in INI-CET November 2022 and May 2022. I chose these 2 exams because these 2 exams had classically a unique type of MCQs. So I've chosen all those MCQs. Using those MCQs, I'll try to cover as many topics as possible. Okay. So good evening, Princey. Good evening, Akhilesh. And before we start the session, let me give you a few facts about Unacademy's plans. So we have something called as Selection Ka Sankalp. So you are lucky that now we are offering you flat 25% off on medical PG subscription. And this would include your access to both live and recorded sessions. And the duration of this course would be 9 months. And the course is aimed next or NEET PG 2024. And this is available at the cost of 17,983 when the actual cost is 23,978. And to avail this offer, you can use any educator's code. If you want, you can use my code, which is C Shanmugapriya. Hi, Titir. Hi, Krishna. And then these are the this is the success story of Unacademy. So all these are Unacademy Plus subscribers who got good score and good rank in the latest NEET PG 2023 exam. And this is about the course. So all of us will be discussing all high yield topics and concepts, and the duration would cover 9 months. Okay, and I'm skipping this because it's about the next batch, and those who are attending this session will not be interested in knowing about the next batch. You're all interested in INI-CET batch, right? So for INI-CET aspirants, we have Mission INI-CET 2023 High Yield Revision batch, and this will be an exclusive rapid revision score booster batch, and we will be covering the latest 3-year previous year questions, and this is available at the cost of 3,599. And to avail this offer, again, you can use my code, which is my name, C Shanmugapriya. Okay. And on popular demand, last mile preparation by Dr. Zainab Bora is also available on the Unacademy too. So that's all about. I can tell you about vitamins. Yes, there are few MCQs on vitamins, but if you want, I can summarize and tell you all about vitamins. Okay. So this is the first MCQ. This is an INI-CET November 2022. What is the diagnosis? So what is that you see here? Do you see, what do you see here? Orange tonsil, right? So this is called as grayish orange tonsils. And how did I ask you to remember grayish orange tonsils? Orange tang. Yeah, orange tang juice. So tang here stands for Tangiers disease. So if you are given an image wherein tonsil is orange color, then that is Tangiers disease. And what are the other facts that you know about Tangiers disease? Tangiers disease is caused by the defect of ABCA1. What is ABCA1? It is ATP binding cassette transporter 1 mutation causes Tangiers disease. And this Tangiers disease is characterized by hepatosplenomegaly. Not only grayish orange tonsils, they are also present with hepatosplenomegaly, mononeuritis multiplex. Okay, so if you know these facts, that's more than enough about Tangiers disease. Now let's try to learn about other choices as well, because it's entire biochemistry in 2 hours, right? So what is that you know about old man's disease? Old man's disease is caused by the defect of acid lipase. And when acid lipase is defective, when you find old man's disease, there is an image based question that can be asked as far as old man's disease is concerned. That is adrenal calcification. So if you see an image like this, yeah, it's an X-ray wherein adrenals have undergone calcification, then what should you suspect? You should suspect acid lipase defect or old man's disease. Porphyria, okay, I will tell you porphyria. Okay, what summary? Okay, I will do you. I will definitely give you a summary of vitamins today. Okay. So that's about old man's disease. Now about Gaucher's disease. We know Gaucher's disease is nothing but beta glucosidase defect, right? It is nothing but beta glucosidase defect or beta glucosyl cerebrosidase defect. So when this enzyme is defective, logically what will accumulate? Glucosyl cerebroside will accumulate in the membranes of RBCs and platelets. And these abnormal membrane RBCs and platelets will be engulfed by macrophages. And that causes, what happens when RBCs and platelets undergo destruction? That will cause anemia and thrombocytopenia. And because of extramedullary hematopoiesis and because of accumulation of glucosyl cerebroside, there is hepatosplenomegaly as well, right? And when macrophages engulf these cells, RBCs and platelets, the membrane which contains beta glucosyl cerebroside will accumulate within macrophages, causing tissue paper cells. Okay, so tissue paper cell is a classical feature of Gaucher's disease wherein cerebrosides accumulate caused by the defect of beta glucosyl cerebrosidase. And what is a probable image-based question that they can ask you as far as Gaucher's disease is concerned? Apart from tissue paper cells, they can also show you Erlen-Meyer-Flass deformity. Have you all heard of it? Yeah, Erlen-Meyer-Flass deformity. So this is a defect of diaphysis of femur. Yeah, so this is how the femur would look, which makes it look like an Erlen-Meyer-Flass. So if you see that, what is your diagnosis? Your diagnosis is Gaucher's disease. And Tay-Sachs disease, what is that? I have already told you about Tay-Sachs disease. Tay-Sachs disease is caused by the defect of hexosaminidase A gene. And the enzyme that is defective in this condition is hexosaminidase A enzyme. And because of this, GM2 gangliosides accumulate. Please memorize all these facts. When hexosaminidase A enzyme is defective, which ganglioside accumulates? GM2 ganglioside accumulates. So Tay-Sachs disease is one of the GM2 gangliosidosis. And this presents with cherry red spot. This condition presents with cherry red spot with predominantly neurological manifestations. So can you tell me how will you be able to differentiate, very good Vinod Jadhav, how will you be able to differentiate GM1 gangliosidosis from GM2 gangliosidosis? In GM1 gangliosidosis, there is not only neurological manifestation, additionally they present with organomegaly, they present with hepatosplenomegaly, they also present with skeletal manifestations. Whereas GM2 gangliosidosis would present only with neurological manifestations, they don't present with hepatosplenomegaly. And Tay-Sachs disease will present as cherry red spots. So the image-based question that can be asked as far as Tay-Sachs disease is concerned is cherry red spot. Have you all understood this? So that's about this MCQ. Now the next MCQ, very good Yuri Gabriel, good. So this is the next MCQ. This is also a previous year MCQ asked in the same INI-CET November 2022. Glycogen phosphorylase is activated directly or indirectly by. Can you all start answering this? Tell me, is it A or B or C or D? Calcium glucose 6-phosphate, insulin. Dr. RJ says it's A. Hi Triple N. My pleasure Triple N. Tithir Surai also thinks it's A. Paramasivam thinks it's C. Okay, he thinks it's insulin. Please do these sessions for physiology. Okay, I will try to do it. So, whenever you're asked about regulation, all of you please listen to this. If the question is about regulation of an enzyme, the first question that you should ask yourself is this enzyme an anabolic enzyme or a catabolic enzyme? Can you do that? Ask yourself, is this an anabolic enzyme or a catabolic enzyme? Why do you ask this question? Because if it's an anabolic enzyme, it will be stimulated by insulin. Whereas if it's a catabolic enzyme, it will be stimulated by glucagon. That way you'll be able to exclude choices quickly. The next question that you should ask yourself is, what are the substrates and what are the products? Why should you ask that question? Because substrates always stimulate an enzyme and products always inhibit an enzyme. So if you know the answers to these two questions, then you'll be able to answer any MCQ on regulation. Let me give you an example. Glycogen phosphorylase is an enzyme of which pathway, all of you? Glycogen phosphorylase is an enzyme of glycogenolysis. Okay. Your conceptual teaching helped in ABG and storage disorders. I can definitely teach few topics in medicine. I can definitely teach you ABG. I will try to do few sessions in medicine. Okay. So glycogenolysis. Now tell me, is glycogenolysis an anabolic pathway or a catabolic pathway? Glycogenolysis is a catabolic pathway. If it's catabolic pathway, will it be stimulated or inhibited by insulin? It is going to be inhibited by insulin. So which choice have you excluded here? It's activated directly or indirectly by, it is not insulin. Got it? Now ask yourself, what are the products of this enzyme? Glycogen phosphorylase directly forms glucose 1-phosphate. This glucose 1-phosphate becomes glucose 6-phosphate, and that in the presence of glucose 6-phosphatase becomes glucose. So what are the products of glycogen phosphorylase? It's glucose 1-phosphate, glucose 6-phosphate and glucose. And all these three being products will inhibit the enzyme. So what are the choices that you have excluded? You have excluded choice B, you have excluded choice D. And what are you left with? You are left with choice A. So glycogen phosphorylase is activated directly or indirectly by calcium. Why should that happen? Because this has to happen in muscle. So in muscle, can you tell me how is excitation, contraction, coupling brought about? No, Tithar, this is not a cofactor. This is covalent modification. Yeah, this is not a cofactor, this is covalent modification. So this should happen in muscle. So in muscle, how is excitation, contraction, coupling brought about? It is brought about by calcium release from the storehouse. And once calcium is released from the storehouses, you know it binds to troponin. And when it binds to troponin, you know myosin head goes and binds to actin. But you know that just myosin head going and binding to actin is not sufficient to bring about the power stroke. Myosin head needs ATP. So once calcium is released from sarcoplasmic reticulum, calcium knows that my purpose is to stimulate excitation, contraction, coupling, for which it is not enough if I just go and bind to troponin. I should also stimulate all the pathways which can provide the necessary ATP. And calcium knows in muscle only following glycogenolysis ATP can be produced, and that is why calcium stimulates glycogen phosphorylase. Do you understand this? Tithar Surai wants me to explain contraction relaxation process. Okay, I will try to tell you. For now, have you understood this MCQ? So whenever you are seeing an MCQ on regulation, it can be about glycogen phosphorylase, it can be about carnitine acyltransferase, it can be about acetyl-CoA carboxylase, that is immaterial. What are the two questions that you will ask yourself? The first question is it an anabolic enzyme or a catabolic enzyme? Second question, what are the products? Yeah, just by that you will be able to answer any MCQ. Okay. How glucuronic acid, eduronic acid? I will tell you, Sukhbir Singh. So epimers, they are examples of spatial isomerism. Right, they are examples of spatial isomerism or stereoisomerism, wherein they differ in spatial orientation of H and OH groups. And in every session, I've told you about epimerism related only to glucose. I've never told you about glucuronic acid, because once it becomes glucuronic acid, even the first carbon atom becomes asymmetric. Okay, and when that becomes asymmetric, there can be epimerism with respect to the first carbon atom also. So in that first carbon atom, when carboxyl group is above the plane of the ring, it is glucuronic acid. If carboxyl group is below the plane of the ring, it is eduronic acid. Do you understand that, Sukhbir Singh? Yeah. That is why glucuronic and eduronic are epimers, and that's why epimerase can do it. Okay, so all the examples that I've given so far in all my sessions is related to glucose. In glucose, the first one is not an asymmetric carbon atom, but glucuronic acid it is. Okay. Burnt sugar, yes, you have to see it. Yes, you're right. Burnt sugar smell of urine is because of defect of what? So this is again a November 2022 INI-CET MCQ. What is the right answer? Burnt sugar smell of urine is nothing but maple syrup urine disease. And what are the facts that you need to know about maple syrup urine disease? It is caused by the defect of, it's caused by the defect of branch chain keto acid dehydrogenase enzyme. And because this enzyme is dependent on thiamine, a patient of maple syrup urine disease will respond to thiamine administration. Okay, so answer for this MCQ is choice D, but let's learn about other choices as well. Tell me, phenylalanine hydroxylase defect. Yes, there are so many questions on molecular biochemistry today, Triple N. Okay, yeah. So phenylalanine hydroxylase defect. Can you tell me the name of the condition? Name of the condition is phenylketonuria. And what did I tell you yesterday in the 2-hour session? Phenylketonuria, keyboard and mouse go hand in hand, right? Keyboard and mouse go hand in hand. So phenylketonuria, keyboard, mouse. So it's characterized by what odor of urine? Mousy odor of urine. Okay, and fumaryl acetoacetate hydrolase defect. This defect causes type 1 tyrosinemia. And type 1 tyrosinemia is characterized by cabbage odor. It is type 2 is what, Titar? I said type 2, 2 T's. It is tyrosine transaminase defect. Okay, type 2 tyrosinemia is caused by the defect of tyrosine transaminase. Fumaryl acetoacetate hydrolase defect causes type 1 tyrosinemia. You can remember an image which I have always showed you. Tire is being repurposed to grow cabbage. Okay, tire is being repurposed to grow cabbage. So type 1 tyrosinemia is characterized by cabbage odor of urine. Okay. Now isovaleric-CoA dehydrogenase deficiency causes isovaleric acidemia. It causes isovaleric acidemia, and this is characterized by sweaty feet odor. How will you remember isovaleric acidemia is characterized by sweaty feet odor? I have always shown you an image wherein feet are kept on the ice. Okay, in that image feet are kept on the ice. So isovaleric acidemia is characterized by what? Sweaty feet odor. Do you all understand this? Okay, I've already seen it. Good. So that's about all these odors caused by the defect of branch chain keto acid dehydrogenase enzyme. C causes which disease? Isovaleric-CoA dehydrogenase deficiency is isovaleric acidemia. It's called as isovaleric acidemia, characterized by sweaty feet odor. Okay. So cystic fibrosis is caused by which of the following deletion? This has been frequently asked in AIMS, frequently asked in INI-CET. So cystic fibrosis is caused by deletion of phenylalanine. Yeah, we call it as delta F508. What do you mean by delta? Delta is deletion. F stands for phenylalanine, and this is the 508th amino acid. Okay, so what is answer? Cystic fibrosis is caused by which of the following deletion? Phenylalanine at 508th position. So cystic fibrosis is an autosomal recessive disorder. It is caused by the defect of CFTR protein. Yeah, what is CFTR? It corresponds to a chloride transporter. And this chloride transporter is responsible for chloride movement in sweat glands. It is responsible for chloride movement in many exocrine glands, okay, and in many secretions. And that is why, you know, features of cystic fibrosis would be sweat chloride will be very high, right? Sweat chloride test will be very high. And then apart from that, because all secretions become thicker, yeah, they present with the obstruction of the respiratory tract or obstruction with thick mucus plug. Obstruction of the pancreatic duct with thick mucus plug. So they present with chronic pancreatitis or pancreatic obstruction, and that causes steatorrhea. Okay, so cystic fibrosis, the most common mutations delta F508, deletion of phenylalanine at 508th position. It is an autosomal recessive condition caused by the mutation of the gene which codes for CFTR protein that is responsible for chloride ion movement in sweat glands, because of which sweat chloride concentration increases. and because it's responsible for making the secretions fluid. When this protein is defective, all secretions become thick and mucus, and that causes respiratory tract obstruction and repeated respiratory tract infections, and causes obstruction of pancreatic duct with thick mucus plug. So it presents with pancreatic duct obstruction and steatorrhea. Okay. So this is also an INI-CET November 2022 MCQ, and I found this question to be a bit difficult one, because if you have not learned HMP shunt conceptually, you will not be able to answer this MCQ. Okay, can you try answering this? The significance of HMP shunt are all except. Is it a source of NADPH? Is it a source of ribose 5-phosphate? Is it a source of acetyl-CoA? Utilizes which of the following is unrelated to DNA methylation? Okay, what is it? 7-methylguanosine cap addition. So 7-methylguanosine cap addition does not happen in DNA. Do you understand this, Yuri Gabriel? Yeah, 7-methylguanosine cap addition happens on the mRNA, and the question was DNA methylation. So I would go for that. And if you're asking me, this DNA methylation related to replication, yes, it is related to replication. So Yuri Gabriel has got a doubt about a 2020 molecular biology MCQ. The MCQ which he has asked about is DNA methylation is not related to. Okay, so what are the effects of DNA methylation? So whenever there is DNA methylation, particularly methylation of cytidine residues, at CG islands, that causes down regulation of the gene. So that part of the chromosome will not be expressed, it becomes heterochromatin. And that was one of the choices. One of the choices was it results in chromatin remodeling. It causes down regulation of gene expression, both of which are true. Okay, it causes chromatin remodeling and it causes down regulation of gene expression. And then choice C and choice D. Choice C was 7-methylguanosine cap addition. So even here there is methylation, but this methylation is happening to DNA, not to, it's happening to RNA, not to DNA. So you can go for choice C. And what was the last choice? The last choice was replication. Is it related to replication? Yes, it is related to replication. Because as there is aging, yeah, whenever there is aging, let me show you here. So whenever there is aging, the DNA will undergo methylation. So suppose this is the template strand. The template strand has undergone methylation, and when this undergoes replication, which will be treated as a template strand, the 3', 5' strand will be treated as a template strand, and the new DNA strand will be synthesized. So do you understand that methylation is related to replication? This is how you identify the template strand. Okay, have I answered your question, Yuri Gabriel? So answer for that MCQ about which I told you is 7-methylguanosine cap addition. Could not understand that in the slide. Okay. So the MCQ which Yuri Gabriel asked is, let me write down the MCQ. DNA methylation. The MCQ is DNA methylation is not related to. Okay, and this is a 2020 INI-CET MCQ. Okay, choice A was chromatin remodeling. Choice B was down regulation of gene expression. Okay, and choice C was 7-methylguanosine cap addition, and the last choice is replication. What is the right answer? The right answer is 7-methylguanosine cap addition, because 7-methylguanosine cap addition is one of the post-transcriptional modification that happens on mRNA. So it's happening in RNA, not on the DNA, and that is why this is the right answer. So is it related to chromatin remodeling? Yes, because whenever there is methylation of cytidine residues of CG islands, So some segments of your chromosome are rich in CG sequences. In those CG islands, if there is methylation of cytidine residues, what happens as a result of this is the chromatin in that region will undergo supercondensation. Once it undergoes condensation, it cannot be expressed. So there will be down regulation of gene expression. So S, that results in chromatin remodeling. S, that results in down regulation of gene expression. It's also related to replication because template strand is identified as that strand which is methylated, because methylation of DNA is a sign of aging DNA. That is how you identify the template strand. Got it? In FMG, what is your doubt? Yes, FMG, there are so many questions which are asked in molecular biology these days. Okay, so don't think that it's not necessary for FMG. It will be essential for FMG, and also if you're an FMG aspirant, any day you will be writing your NEET PG, right? So NEET or next. So no point in worrying about learning lots. This will definitely help you in answering a simple MCQ. Okay, so let's come back to the question that I was asking you about. The significance of HMP shunt or all except. The answer for this MCQ is utilizes ribose. It does not utilize ribose. Do you all understand this? HMP shunt does not utilize ribose. So let me tell you about the other choices. HMP shunt happens in two phases. What are the two phases? The first phase is an oxidative phase. Yeah, the first phase is an oxidative phase. The next phase is a non-oxidative phase. And what is the significance of oxidative phase? As the name indicates, an oxidative phase, you oxidize glucose 6-phosphate. You remove hydrogen atom from glucose 6-phosphate. You give that hydrogen to NADP, forming NADPH. So what is one significance of HMP shunt? It acts as a source of NADPH. Now, what is the significance of non-oxidative phase? It acts as a source of ribose 5-phosphate, and that is why you call HMP shunt as pentose phosphate pathway. So, do you have any doubt about choice A or choice B? The two significances of HMP shunt are they act as a source of NADPH, they act as a source of ribose 5-phosphate. Now, coming on to choice C. The final product of HMP shunt is, in HMP shunt, you start with 3 molecules of glucose 6-phosphate and the end point would be 2.5 glucose 6-phosphate, because the remaining 3 carbon atoms would have come out as carbon dioxide. So always in HMP shunt, you start with 3 glucose 6-phosphate molecules and at the end you will end up getting 2.5 glucose 6-phosphate. Now, what will be the fate of this 2.5 glucose 6-phosphate? They all get into glycolysis. What will happen when they get into glycolysis? You get pyruvate. And from pyruvate, you can get acetyl-CoA. So, HMP shunt can indirectly act as a source of produce. Only little bit confused. I don't understand your point, Paramasivam. Please elaborate. So, indirectly your HMP shunt can act as a source of acetyl-CoA, because the product of HMP shunt would be 2.5 glucose 6-phosphate. But it does not utilize ribose 5-phosphate. And if your question is, if it does not utilize ribose, how will it form ribose 5-phosphate? It is because in HMP shunt, glucose 6-phosphate gets converted to 6-phosphogluconate. Then when 6-phosphogluconate dehydrogenase acts on 6-phosphogluconate, 1 carbon atom is removed as carbon dioxide. So from a hexose, because you have lost 1 carbon atom, what is the product you will get? You will get directly a pentose. And what is the pentose that you get? It is ribulose 5-phosphate. Okay, this ribulose 5-phosphate in the presence of ketoisomerase becomes ribose 5-phosphate. So in any of these steps, do you see ribose to be utilized? No, though it forms ribose 5-phosphate. What is not clear, Paramasivam? You will have to be very specific. What is not clear? I am telling you the significance of HMP shunt. Okay, let me finish this first. So, ribose 5-phosphate is formed in HMP shunt, and for which we are not using ribose. From where do you get the ribose 5-phosphate? You get ribose 5-phosphate from glucose 6-phosphate, because 6-phosphogluconate dehydrogenase releases one carbon atom as carbon dioxide. Okay, yes, until you form ribulose 5-phosphate, you call it as an oxidative phase. After ribulose 5-phosphate is formed, it is non-oxidative phase. Okay, so what is the take home message, all of you, for HMP shunt? I want all of you to answer this along with me. The take home message for HMP shunt is the only pathway wherein ATP is neither utilized nor generated. Okay, so what is the significance of HMP shunt? There are two phases of HMP shunt. One is oxidative phase, the other one is non-oxidative phase. Oxidative phase acts as a source of NADPH. So if you ask which is the major source of NADPH, your answer should be HMP shunt. And the significance of non-oxidative phase is that it acts as a source of ribose 5-phosphate. Okay, and from where do you get ribose 5-phosphate? Directly from glucose 6-phosphate, not from ribose. So ribose is not used. And at the end of HMP shunt, you get 2.5 glucose 6-phosphate that indirectly acts as a source of acetyl-CoA. Okay. Okay, got it. So can you tell me what are the sources of NADPH? Now that we have discussed about NADPH, can you tell me what are the sources of NADPH? This has been frequently asked. Major source of NADPH is HMP shunt. And apart from HMP shunt, it is cytoplasmic isocitrate dehydrogenase. The third one is malic enzyme, which converts malate to pyruvate. So tell me what are the sources of NADPH? Major sources HMP shunt. Apart from that, cytoplasmic isocitrate dehydrogenase and malic enzyme. Refsum's disease is caused by the defect of. So for some reason, INI-CET has got a favoritism towards Refsum's disease. Okay, so Refsum's disease is a paroxysmal. Can you give me a second? Give me a second. I'm sorry, I had to go and check who was on the door. Okay, so what was I telling you? I was telling you about, yeah, so Refsum's disease. Refsum's disease is a peroxisomal disorder. Okay, that is point number 1. And point number 2 is it's caused by the defect of alpha oxidation. It's caused by the defect of alpha oxidation of branched chain fatty acid. So all of you tell me, how do fatty acids generally get oxidized? All fatty acids get oxidized by beta oxidation. The only place where an alpha oxidation happens is when it is branched chain fatty acid. And this is done by phytanic acid hydroxylase. The enzyme is phytanic acid hydroxylase, yeah, which mediates alpha oxidation of branched chain fatty acid, and this enzyme is present in peroxisome. And a defect of phytanic acid hydroxylase will result in Refsum's disease, wherein predominantly you will see neurological manifestations. So what is the right answer here? It is defect of alpha oxidation of branched chain fatty acids. Now this brings another fact to my mind, which I feel you'll have to learn. Okay, so they might ask you, what are the functions of peroxisomes? So tell me, what are the functions of peroxisomes? Can you all answer? The major function of peroxisome is that it is related to very long chain fatty acid oxidation. Okay, so please remember, very short chain, medium chain and long chain fatty acids undergo oxidation in mitochondria. Mitochondria cannot oxidize very long chain fatty acids, and very long chain fatty acids undergo oxidation in peroxisome. And just now I told you peroxisome is related to alpha oxidation of branch chain fatty acid. It is also related to ether lipid synthesis. Peroxisome is also related to ether lipid synthesis. If you're asking me what are ether lipids, you have to know that they are plasmalogens. So irrespective of whether they ask you this question as ether lipid or as plasmalogen, you should be able to tell them that peroxisome is related to ether lipid synthesis or plasmalogen synthesis. The fourth function is it's related to conjugation of bile acids. You know, after bile acids get synthesized, they get conjugated with glycine and taurine, right? So that conjugation of bile acid happens in peroxisomes. Is that clear, all of you? So tell me what are the, yes, complete revision in 2 hours, you're right. So tell me what are the functions related to peroxisome. Peroxisome is related to very long chain fatty acid oxidation, alpha oxidation of branch chain fatty acids, ether lipid synthesis or plasmalogen synthesis. It's related to conjugation of bile acids. Okay. Now, this is an interesting question. For a very long time we were saying, okay, SARS-CoV-2 will be asked in this exam. SARS-CoV-2 will be asked in this exam, but never it was asked. The first time it was asked is INI-CET November 2022. And this was the MCQ. SARS-CoV-2 diagnosis is done by all except. Can you write the answer? Which is the right answer? Paramasivam says it's D. Many of you are saying it is C. Faisal says it's RT-PCR. How will you say it's RT-PCR? The question is done by all except. So let me ask you one question. SARS-CoV-2. Is it an RNA virus or a DNA virus? SARS-CoV-2 is an RNA or a DNA virus? It's an RNA virus. Okay. So if it's an RNA virus, the first step would be to convert RNA to DNA. Which enzyme converts RNA to DNA? That is done by reverse transcriptase. Yeah, do you all know that transcription is DNA to RNA? All of you? Do you know that transcription is DNA to RNA? So what is reverse transcription? It is RNA to DNA. So for diagnosing any RNA virus, what is step number 1? Step number 1 is you have to convert the RNA to double-stranded DNA. Only then you can subject it to any amplification technique like PCR. So the first step is reverse transcription PCR, shortly called as RT-PCR. Okay, so if you have something as RT-PCR, it means it is reverse transcription PCR. All that this can do is it can convert RNA to DNA. Then you have to quantify the DNA. How will you quantify the DNA? Using real-time PCR. So do you understand that RT-PCR and real-time PCR are not synonymous? RT-PCR is reverse transcription PCR. That should be followed by real-time PCR or quantitative PCR, which quantifies a number of nucleic acid of SARS-CoV-2 that is present in the sample. So both these will be involved in SARS-CoV-2 diagnosis. But not always you can involve yourself in molecular diagnostics. Sometimes you want a simpler technique. And that is why we thought, why not we detect the antigen instead of the nucleic acid. And to detect antigen, we need an antigen antibody reaction based assay. And that is why we went on to use immunoassays. So do you all know about antigen based testing? All of you, do you all know about antigen based testing? Yeah, many CARD tests. How many of you have done CARD test? All these CARD tests are antigen antibody based testing. They are immunoassays. Okay, so you know A, B and D, all these can be used for SARS-CoV-2 diagnosis. But what is not used? Southern blotting can never be used. So tell me, what is southern blotting? What are the 3 blotting techniques that you know? Yeah, southern blotting, northern blotting, western blotting, right? Southern blotting, northern blotting and western blot. Antibody test, antigen antibody test. I will tell you about that now, Vinod Jadhav. So the third one is western blot. Okay, so southern blot is for DNA. The first time when this technique, when the blotting technique was used, they used it for DNA identification. So southern blot is named after the person who invented this technique. So southern blot is for DNA. Whereas northern blot is for RNA. Western blot is for proteins. Do you all understand this? Southern is for DNA, northern is for RNA, and western blot is for proteins. And will you be able to use southern blot to diagnose an RNA virus infection? You cannot use it. So southern blotting cannot be used here. Okay, and few of you were asking me about the differences between PCR based assay, RT-PCR based assay, and antigen antibody testing, right? So the basic differences, let me classify it into 3 types: RT-PCR, antigen testing, and antibody testing. Obviously, the first difference is RT-PCR is the most sensitive. That will be the most sensitive. Why will it be the most sensitive? Because here you are amplifying the nucleic acid and then you are quantifying. So even if the person has got like 1 or 2 copies, usually RT-PCR will tell you something called as limit of detection. And the limit of detection for RT-PCR for SARS-CoV-2 will be as low as 10 copies per microliter of the sample. Which means even if the individual has got just 10 copies of nucleic acid of SARS-CoV-2 in his or her swab sample, it is going to be positive. Okay, whereas antigens and antibody, there is no amplification at all. So they are not that sensitive. And that is why ICMR recommends RT-PCR for diagnosing SARS-CoV-2. Otherwise, you are going to end up giving false negative reports. Okay, now between antigen and antibody tests, both antigen and antibody tests will be based on immunoassays. Okay, they'll be based on immunoassays, and the difference between antigen based testing and antibody based testing is obviously the window period. Antigen testing, the window period will be shorter. What do you mean by window period? The time between the entry of the organism and the time at which you will be able to detect the infection. That is window period. So window period for antigen testing will be lower, whereas for antibody to be produced, it takes a longer time. So antibody test, the window period will be longer. So which is the best? The best would be RT-PCR followed by antigen based testing followed by antibody based testing. ELISA is an immunoassay. Yeah, ELISA is enzyme linked immunosorbent assay, right? It is enzyme linked immunosorbent assay. So this is also an immunoassay. And what is ELISA is, here the antibody or the antigen will be labeled with an enzyme. Do you understand this? Here the antigen or antibody will be labeled with an enzyme. Let me show you that here. So ELISA, suppose it's a sandwich ELISA. So what is a sandwich ELISA? So in all these ELISA techniques, you will use wells. Right? What will use? You will use wells. And these wells will be coated with antibody. And what do you call these antibodies in the well as? These are called as capture antibodies. Why do you call them as capture antibodies? When you add the sample, if the sample has got the antigen, if the sample has got the antigen, these antibodies will capture those antigens. Okay, and after you add the sample, after you allow the antigen that is present in the sample to be absorbed by the antibody or capture antibody, what you would do is you'll wash the well and then you add conjugate antibody. What do you mean by conjugate antibody? These are labeled antibodies. Okay, these are labeled antibodies. And what will you label these antibodies with? These antibodies are labeled with enzyme. Okay, so after you allow these are called as what? Conjugate antibodies. What do you mean by conjugate antibodies? These are labeled antibodies. So when you add the substrate for this enzyme, the enzyme will act on the substrate and will give you a color. So if you see a color, that means there is antigen in the sample. And the intensity of the color is directly proportional to the concentration of antigen that is present in the sample. So do you understand about ELISA Tithir? Yeah. So why is it called as ELISA? It is enzyme linked immunosorbent assay. So what are the two antibodies that are used here? We call this a sandwich immunoassay. Why am I calling it a sandwich immunoassay? Because in this case, the antigen is sandwiched between two antibodies. What are the two antibodies? One is a capture antibody. The other one is a signal antibody. Got it? Okay, so this is a type of immunoassay, and depending upon what is the label you use, not always you're going to use enzyme as a label. Sometimes you can use a chemiluminescent label, sometimes you can use an electrochemiluminescent label. So depending upon the label that is used, you have various types of immunoassays, but it's enough if you know just about ELISA. Good. Okay, so can we get on to the next MCQ? Yeah, radio immunoassay, the label will be a radioactive substance, and that is obsolete. These days you're not allowed to label anything with a radioactive substance. So radio immunoassay is obsolete. We don't use any radio immunoassay. We use ELISA or chemiluminescent immunoassay or electrochemiluminescent immunoassay. Yeah, what is the right answer here? Selenocysteine is present in which of the following enzymes? Selenocysteine is present in glutathione peroxidase. Very good. Glutathione peroxidase. So what are the three enzymes which need selenocysteine? It is necessary for glutathione peroxidase. The next one is thioredoxin reductase. The next enzyme is thioredoxin reductase. The third enzyme is 5'-deiodinase. All of you please memorize it. It is glutathione peroxidase, thioredoxin reductase and 5'-deiodinase. All these enzymes need selenium in the form of selenocysteine. So glutathione reductase, what is the significance of glutathione reductase? As I've already told you, if you suspect B2 deficiency, if you suspect B2 deficiency, what is B2 otherwise called as riboflavin deficiency? So if you suspect riboflavin deficiency, ribbon and glue go hand in hand. So it is glutathione, RBC glutathione reductase activity that you can estimate. Okay. So here it is glutathione peroxidase, not glutathione reductase. So true about sirtuins except. How many of you have seen this MCQ? Yeah, true about sirtuins except. So watch this MCQ discussion because I'll help you understand so many practical aspects here. Okay. Sirtuin, how do you abbreviate sirtuin? Sirtuin is nothing but silent mutant type, silent mating type information regulatory protein. Okay, it is nothing but silent mating type information regulatory protein, and these proteins have two functions basically. The first function is ADP ribosylation. You think A is the right answer? No. A is involved. A is one of the activities of sirtuins. So there are so many types of sirtuins. You have sirtuin 1 and sirtuin 6, both of which are involved in improving insulin sensitivity and they support in glucose metabolism. Okay, so answer is D. Okay, you're right. Very good. Yes, answer is D. I'm going to explain that now, and that will help you understand. Yes, sirtuin is present in red wine. You will remember only what you like, is it? Yeah. So of course, consumption of red wine, they say will delay aging. But now I'm going to tell you so many other facts. Listen to this. So what are the two enzymatic activities sirtuins have? The first enzymatic activity is ADP ribosylation. Okay. And the next enzymatic activity is histone deacetylation. So tell me what will happen because of these two enzymatic activities. When you deacetylate histones, histones will retain their positive charges. So they retain their interaction with DNA. So the DNA will remain condensed. So because of the activity, because of these two enzymatic activity, your chromosomes will remain highly condensed. And this is one of the mechanisms by which DNA repair mechanism is brought about. Okay, whenever chromatin remains condensed, there will be less exposure of this chromosome to any mutant. And even if there is a mutation, the repair mechanism quickly happens. So because sirtuin causes chromatin condensation, it supports in DNA repair mechanism. And as long as you keep repairing the DNA, there will be delay in aging. So this is the major mechanism by which it delays aging. But additionally, as I told you, sirtuin 1 and sirtuin 6 support glucose homeostasis by increasing insulin sensitivity. Okay, now have a look at this. Because it stimulates ADP ribosyl transferase, During this conversion, it uses NAD acids coenzyme. What is the coenzyme that is required? NAD. Now, what will happen if you are on a high calorie diet? Watch this carefully. What will happen if you are on a high calorie diet? So, the high calorie will keep converting NAD to NADH, which means what will not be available? NAD will not be available. Without niacinamide, what will happen to the activity of sirtuins? Yeah, without NAD, what will happen to the activity of sirtuin? Sirtuin activity will be suppressed. Okay, so when there is high NADH, what happens? Sirtuin action is suppressed, and that is why fasting delays aging. So how many of you, yes, how many of you know that fasting delays aging? So many of you must have heard of intermittent fasting, right? So intermittent fasting is supposed to delay aging. How does it delay aging? Because it increases NAD. When it increases NAD availability, it stimulates sirtuin activity, and that delays aging. Got it? Okay. So what is the right answer here? Sirtuin increases insulin sensitivity and it supports glucose metabolism. Yes, it is a histone deacetylase and it causes chromatin condensation. Yes, it increases the lifespan. But what happens in niacinamide? In the presence of niacinamide, so whenever NAD gets converted to NADH, there is a decrease in the activity. And during the activity of sirtuin, NAD becomes niacinamide. So niacinamide is almost like a product. So niacinamide inhibits sirtuin activity. So D is a false statement. Do you all understand this? D is a false statement. So that's about this MCQ. Okay. So I hope you have understood all about sirtuins. Now choose the right sequence of steps in the conversion of cholesterol to testosterone. This is also an INI-CET November 2022 MCQ. How many of you would have anticipated that such questions can be asked? Don't you think this is a very complex MCQ? Yeah, you are expected to know the sequence of steps involved in the conversion of cholesterol to testosterone. And why are you expected to know the sequence? You will have to know the sequence because only then you will understand about all congenital adrenal hyperplasias. So now I am going to use this chance to help you understand the steps involved in the conversion of cholesterol to all steroid hormones, number 1. And as we learn about the conversion of cholesterol to steroid hormones, I will help you understand what are the C21 steroids, what are the C19 steroids, and what are the C18 steroids, because that also keeps getting repeated. Okay, so tell me, what is the first step in the conversion of cholesterol to testosterone or to any steroid hormone? So the first step is you take cholesterol. I'm going to write down everything. Please, all of you, watch this and memorize. Cholesterol is a C27 steroid. In the sense, cholesterol has got totally 27 carbon atoms. When this cholesterol is acted upon by the first enzyme, the first enzyme is cholesterol desmolase. Okay, the first enzyme is cholesterol desmolase, which removes carbon atoms and it converts cholesterol to pregnenolone. Okay, so what is the product that you get? You get pregnenolone. How many carbon atoms are removed? It removes 6 carbon atoms. That is how you get pregnenolone. So what kind of a steroid is pregnenolone? Pregnenolone is a C21 steroid. This pregnenolone is acted upon by 17-alpha hydroxylase. So the next enzyme is 17-alpha hydroxylase, which converts pregnenolone to 17-hydroxypregnenolone. The next enzyme is 1720 lyase. And this 1720 lyase removes 2 carbon atoms and that converts 17-hydroxypregnenolone to DHEA. And this DHEA is the first male sex hormone that is synthesized. And when DHEA gets converted to androstenedione, then this androstenedione becomes testosterone and that becomes dihydroxytestosterone. And all these are formed after the removal of 2 carbon atoms from C21 steroid. So DHEA, androstenedione, testosterone, dihydroxytestosterone, all these are C19 steroids. Do you all? Zona glomerulosa, zona fasciculata, zona reticularis. So all these 3 zones will take up cholesterol. The first enzyme is cholesterol desmolase, and all the three zones, the first enzyme to act will be cholesterol desmolase, which removes 6 carbon atoms and it converts cholesterol to pregnenolone. So pregnenolone is a C21 steroid. The next enzyme is 17 alpha hydroxylase, which forms 17 hydroxypregnenolone. And then you have 1720 lyase, which removes 2 carbon atoms. That is how you get DHEA. So DHEA is a C19 steroid, which means all male sex hormones. DHEA, androstenedione, testosterone, dihydroxytestosterone, or C19 steroids. Now, how will you convert these androgens to estrogens? To convert all these androgens to estrogen, the enzyme that is used is aromatase. And this aromatase removes 1 carbon atom. So all estrogens, what are the estrogens? We have estrone, okay, estradiol, estrone, estradiol, estriol. All these are C18 steroids. Male sex hormones are C19 steroids. All estrogens are C18 steroids. Now, how about the remaining steroid hormones? What are the remaining steroid hormones? You get progesterone, right? We get progesterone, aldosterone, cortisol. Okay, progesterone, aldosterone, cortisol. All these are derivatives of pregnenolone. So, all these are C21 steroids. Do you all understand this? So, can you tell me what are the C21 steroids? C21 steroids will include progesterone, the mineralocorticoid aldosterone, the glucocorticoid cortisol. All male sex hormones, DHEA, androstenedione, testosterone, dihydroxytestosterone, or C19 steroids. All estrogens, estrone, estradiol, estriol, or C18 steroids. Got it? Okay. So now will you be able to answer this MCQ? I'm skipping all this because I've already told you this. So tell me, choose the right sequence of steps in the conversion of cholesterol to testosterone. What is the first enzyme I told you? I said the first enzyme is a desmolase. Okay, and desmolase will be acted upon by 3-beta hydroxysteroid dehydrogenase. So what I told you is cholesterol becoming pregnenolone. Okay, then I told you 17 alpha hydroxylase, and then I told you 1720 lyase, which forms DHEA. This DHEA should get converted to androstenedione. Okay, so there are 2 ways by which you can synthesize androstenedione. One is through this route. Yeah, one is through this route. The other route is when 3-beta hydroxysteroid dehydrogenase acts on pregnenolone, that becomes progesterone. And when this progesterone is acted upon by 17 alpha hydroxylase, it becomes 17 hydroxyprogesterone. When 1720 lyase acts on it, directly it can become androstenedione. So it gets converted to androstenedione either through this route or through this route. So the first enzyme is desmolase. Okay, the first enzyme is desmolase. The next enzyme is 3-beta hydroxysteroid dehydrogenase. And then you have 17 alpha hydroxylase, and finally it is 1720 lyase. Okay. So answer is choice A. So probably I will do a shorts, okay, a short video on congenital adrenal hyperplasia. How do you find out which type of congenital adrenal hyperplasia it is, and I will post it. Probably that will help you to understand it better. Okay. So this is an interesting MCQ again asked in INI-CET. 2,3 BPG causes unloading of oxygen to tissues because of what? 2,3 BPG causes, do you all know that 2,3 BPG reduces the affinity of hemoglobin for oxygen and it helps in unloading oxygen to tissues? Okay, so the question is, how do you think 2,3 BPG causes unloading of oxygen to tissues? What is the right answer? Many of you, very good. Many of you are saying that it binds to the central cavity of hemoglobin surrounded by histidine and lysine, and it favors salt bridge formation. That is a true statement. Okay, so let's try to understand about 2,3 BPG, and let's try to understand about how hemoglobin and myoglobin work basically. Okay, so listen to this. At the end of this discussion, I will be showing about oxyhemoglobin dissociation curve and how to use that curve and how to interpret the curve. Okay, so hemoglobin structure. What is hemoglobin? Hemoglobin has got one heme in the center that is surrounded by one globin chain, right? And it has got 4 such copies. Hemoglobin has got 4 such copies of heme that is present in the center surrounded by a globin chain. And what is heme? Heme has got a metal in the center. What is a metal that is present in the center? It is Fe2 plus. And this ion in the center, Fe2 plus in the center, is surrounded by a tetrapyrrole ring. So suppose this represents the plane of the tetrapyrrole ring, Fe2 plus will be in a plane below the plane of the ring. Okay, and this Fe2 plus is covalently attached to one amino acid of the globin chain. To which amino acid is it attached? Fe2 plus is attached to histidine F8. Okay, and when this globin chain surrounds the heme, when it comes to the other side, the amino acid which is present on the other side of Fe2 plus is histidine E7. So have you understood the basic structure of hemoglobin? Hemoglobin has got 4 copies of heme in the center surrounded by globin chain. What is heme? Heme has got a metal in the center which is Fe2 plus that is surrounded by 4 pyrrole rings. Suppose this represents a plane of the 4 pyrrole rings. Fe2 plus is in a plane below the plane of the ring. And this Fe2 plus is covalently attached to histidine F8. When the globin chain surrounds the heme, when it comes to the other side, it is histidine E7. Now you have 4 such copies of heme plus globin. When you have 4 such copies, it encloses a diamond-shaped cavity in the center. Okay, what are the 4 globin chains? It is alpha 1, alpha 2, beta 1, beta 2. Okay, and the central cavity is surrounded by parts of globin chain which provide you with positively charged amino acids. What are the positively charged amino acids? Histidine H21, lysine and valine. So the central cavity is surrounded by parts of beta globin chain which provide you with positively charged amino acids like histidine H21, lysine and valine. Okay, and what about 2,3-BPG? 2,3-bisphosphoglycerate, right? 2,3-bisphosphoglycerate is negatively charged. So there is an electrostatic attraction between positively charged central cavity and a negatively charged 2,3-BPG. So this 2,3-BPG sits in the central cavity and it favors salt bridge formation. It is positively 2,3-BPG is negatively charged. It is surrounded by positively charged globin chains. So it favors salt bridge formation. That favors TOT structure. TOT structure favors deoxygenation. So as far as hemoglobin is concerned, there are 2 structures which are possible for hemoglobin. One is taut structure, the other one is relaxed structure. Taut structure is like you squeezing out oxygen. What do you mean by taut structure? If there is a high attraction between individual globin chains, that's called as taut structure. Taut structure favors deoxygenation and relaxed structure favors oxygenation. Okay, when 2,3-BPG sits in the central cavity, it favors salt bridge formation and the salt bridge formation favors taut structure. Taut structure favors deoxygenation. That is how 2,3-BPG causes unloading of oxygen to tissues. Is that clear to all of you? So all of you tell me, how does 2,3-BPG help in unloading oxygen to tissues? Because 2,3-BPG binds to the central cavity of hemoglobin which is surrounded by histidine, lysine and valine, and that favors salt bridge formation, that favors taut structure, that favors deoxygenation. Okay. Identify the left curve given the fact that the right curve is of adult hemoglobin. So first tell me, what is the curve here? The curve here is oxyhemoglobin dissociation curve, right? So in this curve, one curve, the blue color curve is the right curve which is sigmoidal, right? Yeah, the question has got the answer. Do you see that here? Yeah, the question has got the answer. They've given you oxygen dissociation curve of myoglobin. So one curve, the right curve is sigmoidal. Sigmoidal curve is adult hemoglobin. The other one is hyperbolic curve. Hyperbolic curve is of myoglobin. So the right answer here is myoglobin, okay? But I'm going to help you understand oxyhemoglobin dissociation curve better, okay? So how is oxyhemoglobin dissociation curve drawn? What is drawn along x-axis and what is drawn along y-axis? Yeah, no cooperative binding in myoglobin. So along y-axis, don't you think y and percentage look similar? Yeah, y and percentage look similar. So along the y-axis, you have percentage saturation, and along x-axis, you have partial pressure of oxygen. Okay, so this is how a graph is drawn, and in this graph along y-axis you have percentage saturation. As you can see here, it is drawn till 100 percentage, and along x-axis it is partial pressure of oxygen. And oxyhemoglobin dissociation curve is sigmoidal. Okay, and in such a graph you're always worried about whether the curve gets jaldi padao. Yeah, that's what I'm doing. I cannot go any faster than this. Okay, so there is an ideal speed in which every educator teaches you. Yeah, that's the educator style. Yeah, if you watch that video, if you watch the session in that pace, you will understand it much better. Will you all agree with me if I say you cannot be in a hurry for something which has to be long lasting? Triple N, can you tell me yes or no? You cannot be in a hurry for something which is long lasting. So it's not just about the number of facts that you learn. It's not just about the number of concepts that you learn. It's all about the retention. It's all about being able to apply. Okay. So at the end of this session, 2-hour session, you would have understood definitely 100 concepts and more. You would have definitely learned more than 200 facts. Okay. So that's my assurance to you. So don't ask me to hurry here. Yeah. Please watch the session in my own. Yeah. Everybody knows that there are 30 days which are left. And there is no point in hurrying and not being able to answer the MCQ. Okay. So watch this. This is a sigmoidal curve for hemoglobin. And in such a graph, you're always worried about the position of the curve. You always want to know whether the curve gets shifted to the right. This is called as right shifting. Or the curve gets shifted to the left. Because the shift of the curve to the right or the shift of the curve to the left will tell you about P50. So tell me, what is P50? P50 is nothing but partial pressure of oxygen at which it is 50% saturated. So this is 50% saturation. From here, if you draw a horizontal line and you draw a vertical line, this is P50. So what is P50 for adult hemoglobin? It is close to 25. So P50 of adult hemoglobin is close to 25 millimeters of mercury. Whereas what does it mean when I say the curve gets shifted to the right? It means the partial pressure of oxygen at which it is 50% saturated is higher. So whenever you say that the curve gets shifted to the right, PDF will be shared in the Telegram group, Vishwas. Okay. So when you say the curve gets shifted to the right, you mean to say that the P50 is higher. When you say the curve is shifted to the left, you mean to say the P50 is low. So can you tell me, what does low P50 mean? Low P50 means even at such a low partial pressure of oxygen, it is 50% saturated, which means its affinity is higher. So P50 and affinity are inversely proportional. So tell me, what does shift of the curve to the right mean? Shift of the curve to the right means P50 is high and affinity is low. Shift of the curve to the left means P50 is low and affinity is high. Okay, so when you say oxymyoglobin dissociation curve is hyperbolic, you actually mean to say that the curve of oxymyoglobin dissociation curve is shifted far towards the left. What does shift of the curve to the left mean? It means its P50 is very low. So for myoglobin, P50 is 8 millimeters of mercury. For adult hemoglobin, it is 25. For adult hemoglobin, it is 25. For myoglobin, it is just 8 millimeters of mercury, which means even when the partial pressure of oxygen is as low as 8 millimeters of mercury, your myoglobin will not give off its oxygen. Because of that, you cannot use myoglobin for transport. Do you all know that hemoglobin is used for oxygen transport, whereas myoglobin is used for storage? Why can't you use myoglobin for transport? It is because P50 of myoglobin is just 8 millimeters of mercury, which means even at a partial pressure of oxygen of 8 millimeters of mercury, a myoglobin will not unload oxygen to tissues. So you cannot use myoglobin to transport. You can use myoglobin only for storage. Does that clarify all your doubts about oxyhemoglobin dissociation curve? Yeah. So when you're asked when will the curve get shifted to the right, you will shift the curve to the right whenever you want effective deoxygenation to happen to the tissues. Because shifting to the right means P50 is high or affinity is low. When the affinity is low, it means effective unloading of oxygen will happen to the tissues. When will you want effective unloading of oxygen to happen to the tissues? Whenever there is hypoxia, right? Whenever there is anemia. In hypoxia, in anemia, in increase in body temperature, and in acidosis. In all these conditions, these are signals that your tissues need more oxygen. You have to deliver more oxygen to tissues. So there you shift the curve to the right. When will you shift the curve to the left? When you want your hemoglobin to exhibit higher affinity. When will you want your hemoglobin to exhibit higher affinity? Exactly in contrast to this. So whenever there is decrease in body temperature or whenever there is alkalosis, the curve will get shifted to the left. Got it? Good, Triple N, you're right. Okay. So tell me, myoglobin dissociation curve is what shown here. And what is the P50 for hemoglobin? P50 for hemoglobin is 25. P50 for myoglobin is 8 millimeters of mercury. So answer this. This is also an INI-CET November 2022 MCQs. In November 2022, there were 18 MCQs in INI-CET. Okay. So phenylketonuria, assertion, phenylketonuria is characterized by mousy odor. True or false? Keyboard, mouse go hand in hand. So assertion is true. Reason, tyrosine and norepinephrine are not formed in phenylketonuria. Is that true? Yeah, tyrosine and norepinephrine are not formed in phenylketonuria. So what is phenylketonuria? Phenylalanine hydroxylase deficiency. Yeah, very good. Very good, Triple N. So phenylalanine hydroxylase is deficient, wherein phenylalanine cannot be converted to tyrosine. Without tyrosine, you cannot form neurotransmitters because it is the tyrosine which gives rise to dopamine. That gives rise to norepinephrine. That gives rise to epinephrine. So in phenylketonuria caused by phenylalanine hydroxylase deficiency, because tyrosine is not formed, norepinephrine, epinephrine, nothing can be formed. So both assertion and reason are true. But is it because norepinephrine not formed, there is mousy odor in phenylketonuria? No. What is the reason for mousy odor in phenylketonuria? Even yesterday in the 2-hour session I told you, the reason for mousy odor in phenylketonuria is phenyl acetate. As Triple N, or who answered it? Arun Sharma. As Arun Sharma has answered, yes. Phenyl acetate is the reason for phenylketonuria. So what is the right answer here? Assertion is true. Reason is also true. But reason is not the correct explanation for assertion. So what is the right answer here? The right answer here is choice B. Got it? Okay. So quickly about phenylketonuria caused by phenylalanine hydroxylase deficiency. In this case, the accumulated phenylalanine gets converted to phenylketones. So phenylalanine on transamination forms phenylpyruvate. So phenylpyruvate is the first phenylketone that you form. Pyruvate on reduction becomes phenylactate. Phenylpyruvate becomes phenyl acetate. Okay. So what are the manifestations? When there is no tyrosine, tell me. No problem, Triple N. When there is no tyrosine, what will not be formed? Yeah, when there is no tyrosine, melanin cannot be synthesized. Without melanin, there will be hypopigmentation. So what is one manifestation of phenylketonuria? It is hypopigmentation. Okay, and without phenylalanine hydroxylase, when phenyl ketones accumulate, these phenyl ketones will answer ferric chloride test. So the second finding is ferric chloride test will be positive. And these phenyl ketones will compete with neutral amino acids to cross the blood-brain barrier. So brain development gets affected. That causes mental retardation. Okay, and phenyl acetate is the reason for mousy odor. So what are the manifestations of phenylketonuria? Quickly tell me. Phenylketonuria presents with mental retardation. It presents with mousy odor. It presents with hypopigmentation. Ferric chloride test will be positive and Guthrie's test will be positive. Yeah, Guthrie's test. Thanks, Triple N. So the next one is Guthrie's test. So please remember, these two are the tests. These two are the screening tests that can be done to detect phenylketonuria. And what is the reason for mousy odor? It is phenyl. नहीं हो रही मैम, खुद से क्या करें बताइएगा, पढ़ाई नहीं हो रहे हैं. Okay, so the major reason for why you are all not able to read in the last minute, or I would not say it's last minute, you have one more month to go. By this point of time, the major reason for why your productivity is reduced is your anxiety. Yeah, the major reason is your anxiety. Please don't be anxious. Please don't be in a hurry. There is no point in being anxious, okay? Because the result or the success, yeah, the success will be a product of whatever you're doing now. So don't worry about the mark. Don't worry about the rank. It is all about using your time productively. Okay, so frame a schedule for yourself. Write as many grant tests as possible. When I say grant test, you don't have to wait for a platform to give you a grant test. Take a previous year question paper of INI-CET. Start working it out. Yeah, spend 3 hours. Spend the entire 3 hours. Be mindful when you write the test. Okay, so don't be in anxiety. Okay, those 3 hours are not a waste of time. Those 3 hours will help you in multiple ways. So write a grant test, work out those grant test paper. Okay, understand your deficiencies. Yeah, understand where you go wrong, understand why you have not understood few concepts. So learn every MCQ that is asked in that paper. Okay, so it has to be a productive utilization of time. It's not just about a quicker reading. It's not just about learning multiple facts in a limited period of time. Do you all understand this? Yeah, please don't be anxious, because already your bread and butter are assured. Yes or no? Already you're a doctor, right? So your bread and butter are assured. You are never going to say, I don't have my meal, I don't have my next meal. That is assured. So whatever you're doing now is for the betterment. You're just trying to supersede your abilities. You're trying to do something, okay? So don't worry about the INI-CET mark or INI-CET rank. Just keep working out. It has to be a smart work. It has to be hard work put together. The ultimate secret for success is being mindful during your preparation. Yeah, every minute counts. I agree, every minute counts, but how you use that minute matters. Okay, so in that minute, even if you learn just only half of the fact, that's good enough. Yeah, so don't be harsh on yourself. Pat yourself for every small success that you achieve. Even if you finish reading one topic in 1 hour, pat yourself. Okay, I've learned this new topic for today. Okay, that's going to be of use in my practice, even if I don't get that MCQ. Okay, if you do that, you will definitely be happy. Please don't be last attempt family has allowed. Okay, so if this is the last attempt that your family has allowed, there is always the next NEET PG. The next NEET PG, you can get definitely any of the course that you want. Okay, so there are so many courses that are available. There are so many seats that are available. It's just that you're fixed on a course. You're just fixed on a college. Please have an open mind. Not everybody should get into radio diagnosis to be successful in life. Yeah, I would like to reiterate this multiple times. Not everybody should get into radio diagnosis. Not everybody should get into AIIMS to be successful in life. That is just one of the tools that you need to be successful. Okay, there are so many other tools which you effectively use. You will be able to succeed. You will be able to shine, provided you do everything mindfully. Okay, so please don't worry. FMG exam, what we want to do, give some tips, ma'am, and how many hours you have to study. Okay, so be it FMG or NEET PG or INI-CET, it's about the maximum number of hours that you will be able to put in. Okay, and for all of you, I would say minimum 12 hours a day you should devote in reading. Yeah, I might sound to be a very ambitious person, but you have to be ambitious in the number of hours that you put in. Okay, you have to be ambitious not in the number of facts that you learn, in the number of hours that you put in. Okay, so learn conceptually, do your hard work, do your maximum effort that you can put in. And as far as FMG is concerned, you will have to know something about everything. Do you understand this? You will have to know something about everything, because FMG question paper is rapidly evolving. So don't think if I know few topics here and there, I will be able to clear the exam. No, that is not how it works. Okay. So learn all 19 subjects, give equal importance to all. When I say equal importance, the weightage should be had in mind. So you should fix the number of hours for every subject and limit to those number of hours. So the success depends upon the schedule that you put for yourself. So sit today, frame a schedule for yourself, stick to the schedule, be mindful, don't be anxious, pat yourself for every success that you get, okay, and don't be worried much. Okay, you will be successful in one day. Okay, can you say for FMG? That's what I said just now. 6 hours, please, triple N. It's not about watching something in 2x and limiting your number of hours to 6 hours. Extend it. Yeah, extend it. Your brain has got unimaginable potential. Your brain has got unimaginable potential. It's us who's restricting our brain's abilities. Okay, so please extend the number of hours that you devote for learning every day and learn it conceptually. Okay, can we go ahead with the discussion? Consistency nahi aa rahi hai. So it's all about discipline, triple N. You should fix the time. You should fix the time for you to wake up. You should fix the time for when you're going to sleep. And in between this, you should fix the nap time. And apart from these sleeping times, okay, you have to read. You have to read. Okay, so of course, in between, you can treat yourself with like 10 minutes of Instagram or 15 minutes of Instagram reels. But that should not occupy the major proportion of your day. Got it? Yeah. Consistency is in your hands, triple N. So fix the duration that you're going to spend for reading on a day and stick to it. Okay. So let's go back to this. What is the answer here? Yes, assertion is true, reason is true, but reason is not the correct explanation for assertion. So the answer is choice B. Yeah, I will do a rapid revision for FMG for sure, Paramasivam. Don't worry. Long-term glycemic control. How many of you have seen this MCQ? Long-term glycemic control in a person with hemolysis is assessed by estimating fructosamine. Fructosamine is सारा time सोने का मन करती है. When medicos will get time to relax, ma'am. After your exam, you can relax heartily, triple N. You will get enough time to relax. Okay, don't worry about it. Only gifted people get into medicine. So don't be harsh on the choice that you made. You have taken the right choice because you're going to feel gratified every day of your life. There is no other profession. There is no other profession that will give you as much gratification as what you can expect in a medical field. Okay, so don't feel bad about this course. Don't feel bad about this profession. You will feel good. You will never repent after you get into active practice. Okay, yeah. So what is the right answer? It is choice D. Fructosamine is a protein. And what is fructosamine? It is glycated albumin. Fructosamine is nothing but glycated albumin. Okay. So first let me tell you about glycated hemoglobin. So what is glycated hemoglobin? Glycated hemoglobin is non-enzymatic. It is non-enzymatic addition of carbohydrate residues to N-terminal valence of hemoglobin. So in the absence of enzyme, just because there is excess carbohydrate residue in the circulation, some of the N-terminal valence of hemoglobin will undergo glycation. So this is directly proportional to the carbohydrate concentration in the circulation. There is no other factor which determines how much of hemoglobin has been glycated. It's directly proportional to the carbohydrate residue concentration in the circulation. And once hemoglobin undergoes glycation, it is going to be there for its lifetime. And what is the lifespan of RBCs? It is 120 days. So once glycate is going to be there for all the 120 days. So if you measure the proportion of the hemoglobin, total hemoglobin in the denominator, glycated hemoglobin in the numerator expressed as a percentage. If you measure this, that is going to tell you the long-term glycemic control of the individual over the last 2 to 3 months. So it is between 2 to 3 months, in the sense it's 8 to 12 weeks. It is going to give you the glycemic control for the last 8 to 12 weeks. But what is the disadvantage of you estimating glycated hemoglobin in few clinical conditions? For example, if the patient that you are estimating glycated hemoglobin for is suffering from hemolytic anemia. If the person has got hemolytic anemia, what will happen to lifespan of RBCs? Lifespan of RBC will be reduced. That is rapid turnover of RBC. So lifespan of RBC is reduced. So the probability that the hemoglobin undergoes glycation is reduced. So that will give you a false low glycated hemoglobin concentration. That will give a false view that your patient is in long-term glycemic control, which is actually not the case. Do you understand this? So if a person is suffering from hemolytic anemia, that can give you false low glycated hemoglobin concentration. Whereas what happens if the person is suffering from iron deficiency anemia? In iron deficiency anemia, the turnover of RBCs is reduced. So one RBC stays for longer than 120 days. So the probability that this RBC gets exposed to glucose is more. So that gives you false high values. So in hemolytic anemia, there is false low glycated hemoglobin concentration. In iron deficiency anemia, there is false high glycated hemoglobin. And that is when you have to estimate something else to assess the long-term glycemic control, which is fructosamine. And what is fructosamine? It is glycated albumin that is not going to be affected by RBC turnover. But it is going to be affected by the fact that albumin lifespan is not as long as 120 days. Albumin lifespan is shorter. So if you are estimating fructosamine, you will get to know the glycemic control of the individual for not to 8 to 12 weeks. Less than that, it is between 2 and 3 weeks glycemic control. Got it? Yeah, so that's about fructosamine. So tell me, what is fructosamine? It is glycated albumin. And please remember, glycated hemoglobin, there is another MCQ which is being repeatedly asked. Which is the most common method that is used to estimate glycated hemoglobin? It is ion exchange chromatography. What is it? It's ion exchange chromatography, which is the most commonly used technique for estimating glycated hemoglobin. To be more specific, it is cation exchange chromatography. Okay, but apart from chromatography, you also have electrophoresis to estimate glycated hemoglobin. You also have immunoassays. But if the question is which is the most commonly used technique, then the answer is ion exchange chromatography. What? When did I say ulta? I never said hemolytic anemia. Hemolytic anemia, there will be false low value. What did I say? And in iron deficiency anemia, there will be false high values triple N. So this is the right answer. If I had changed previously, then this is the right thing. Okay. So long-term glycemic control in a person with hemolysis is assessed by estimating fructosamine. Fructosamine is what? It is a protein. And glycemic control for what? 2 to 3 weeks. I said correct, right? Thank you, Titir. Which of the following tissues are independent of insulin for glucose uptake? Which is not dependent on insulin for glucose uptake? Can you tell me the right answer? Liver, muscle, adipose tissue, placenta. Is it liver or muscle or adipose tissue or placenta? Do you have any doubt about liver? Do you have any doubt about muscle, adipose tissue? I hope you don't have any doubt about muscle and adipose tissue, because insulin dependent glucose uptake happens through GLUT4 transporters. And these GLUT4 transporters are present in skeletal muscle, cardiac muscle, and adipose tissue. Okay, and adipose tissue. So we know for sure muscle and adipose tissue are dependent on insulin. But liver is also dependent on insulin, the reason for which I will tell you now. But before I tell you the reason, I want you to know in and out about GLUT transporters. The first fact that you should know about GLUT transporters is that they belong to facilitated passive diffusion mechanism. All that your GLUT transporter can do is, it can transport glucose from a region of higher concentration to a region of lower concentration. That is why I say it's an example of passive diffusion. To be very specific, it is facilitated passive diffusion because it uses a carrier protein. Now about location of these transporters, GLUT1 and GLUT3 transporters are present in those cells for which glucose is indispensable. So tell me, what are the cells for which glucose is indispensable? Neurons, right? Neurons, RBCs and placenta. So GLUT1 and GLUT3 transporters are present in neurons, RBCs and placenta. What does that tell you about GLUT1 and GLUT3 transporters? Their affinity for glucose is higher. So that even when there is low plasma glucose, neurons, RBCs and placenta will continue taking up glucose. Okay. Now GLUT2 transporters are present in those cells. Brain stopped working. Okay, good. Good that you have now understood what happens when you watch it in 2x speed. You have to always apply a mind, think and watch the video. Okay. So GLUT2 transporters, these are present in those tissues which are related to glucose balance or glucose homeostasis. So tell me, what are the cells which are related to glucose balance? First, glucose has to be absorbed by enterocytes, right? Then glucose has to be stored in hepatocytes. And pancreatic beta cells should release insulin. And then proximal convoluted tubular cells should reabsorb glucose. So tell me, what are the cells which are related to glucose balance? Glucose balance is related to enterocytes, hepatocytes, pancreatic beta cells and renal tubular cells. And in all these cells, you have GLUT2 transporters. GLUT4, just now I told you, insulin dependent glucose uptake happens in those tissues wherein glucose can be used for anabolism. And insulin knows that if I allow glucose to be taken up by skeletal muscle or by cardiac muscle, that glucose can be stored for glycogen synthesis. And if I allow glucose to be taken up by adipose tissue, that glucose will be stored as fat. And that is why insulin dependent glucose uptake happens in skeletal muscle, cardiac muscle and adipose tissue by GLUT4 transporter upregulation. Okay. And GLUT5 transporter is related to fructose absorption. So have you understood this? Yeah, have you memorized it? So that's about the presence of GLUT transporters. Now tell me, in enterocyte, which transporter did I tell you is present? Which transporter did I tell you is present in enterocyte? Just now I told you enterocytes are related to glucose balance, right? And in glucose balance cells, it is GLUT2 transporter. Now my question to you is, in enterocyte, where do you find these GLUT2 transporters? Do you find GLUT2 transporters along the apical side or along the basolateral side? You have GLUT2 transporters along the basolateral side. So what is present along the apical side? Along the apical side, you have sodium glucose cotransporter 1. Okay, along the apical side, you have sodium glucose cotransporter 1. Why should you know this? Only then you will understand why do you add glucose to oral rehydration solution. This was recently asked. Why do you add glucose to oral rehydration solution? The purpose of you giving oral rehydration solution to any person is to replenish the sodium loss. And to replenish the sodium loss, you should have added just the table salt. But what is that we do? We also add sugar to it. Why do we add sugar to it? Not just to make it palatable, but because we know when I add sugar, when glucose gets absorbed along the apical side, along with that sodium will also get absorbed. I know adding sugar will facilitate sodium absorption. That is why in oral rehydration solution, we add both sugar and salt. So if you remember this, you will know that along the apical side, you have sodium glucose cotransporter 1, and along the basolateral side, you have GLUT2 transporters. Okay. My next question to you is, what about liver? Do you think liver glucose uptake is dependent on insulin? I said liver also has got GLUT2 transporter, and you know GLUT2 transporter is not dependent on insulin. Still, liver glucose uptake is dependent on insulin indirectly, because liver has got an enzyme glucokinase. Yeah, liver has got an enzyme glucokinase, and that is inducible by insulin. So can we learn about the differences between hexokinase and glucokinase now? How many of you want to learn it? Yeah, the differences between hexokinase and glucokinase. What are the differences? The first difference is location. So tell me, where is hexokinase present? Hexokinase is present in all the cells. There is glucokinase is present in liver and pancreatic beta cells. Do you understand this? Hexokinase is present in all cells. Glucokinase is present in liver and pancreatic beta cells. The second difference is based on affinity and Km. Like what I told you about P50 and affinity, Km and affinity are also inversely related. Okay. Now listen to this. Glucokinase, the enzyme which I told you is present in liver and pancreatic beta cells, exhibits lower affinity. It exhibits lower affinity or higher Km for glucose. Why do you want an enzyme with lower affinity to be present in liver? Can you tell me? Why do you want an enzyme with lower affinity to be there in the liver? Because liver is the first organ to meet your dietary glucose. What happens to any diet that you take? Any carbohydrate that is present in the diet gets digested. It gets absorbed into the portal vein. Through portal vein, every dietary glucose reaches the liver first. So can you tell me what would have happened if liver was provided with an enzyme with higher affinity for glucose? Liver would have used all the dietary glucose for its own, so that peripheral tissues suffer from hypoglycemia. You don't want that to happen. That is why nature has provided your liver with an enzyme which can act on glucose only when the glucose concentration is very high. Only when the glucose concentration is more than what is required for the peripheral tissues. Only when there is excess glucose, liver enzyme will be able to act on glucose. That is why glucokinase with lower affinity is present in liver. Now, why do you want an enzyme with lower affinity in pancreatic beta cells, all of you? Why do you want an enzyme with lower affinity to be there in pancreatic beta cells? Because in pancreatic beta cells, it is related to insulin release. Can you tell me what is the sequence of events which happen which lead on to insulin release? Glucose should enter into pancreatic beta cells. Glucose should go through glycolysis and citric acid cycle. As a result of glycolysis and citric acid cycle, you get ATP. This ATP closes ATP sensitive potassium channels. And this closure of potassium channel causes release of insulin. Do you understand this? Closure of potassium channel will cause depolarization and action potential. That action potential causes insulin release. So can you tell me what would have happened if pancreatic beta cells are provided with an enzyme with higher affinity for glucose? If the enzyme in beta cell was provided with higher affinity, this sequence of event would have happened even when there is low glucose. So even when there is low glucose, insulin will be released. Do you want that to happen? Even when there is low glucose, if insulin is released, that will aggravate hypoglycemia, which is dangerous. We want insulin to be released only when there is hyperglycemia. So you want an enzyme with lower affinity to be there in the pancreatic beta cells. So have you understood the differences between hexokinase and glucokinase, all of you? Yeah, what are the differences? Hexokinase is present in all cells. Glucokinase is present in liver and pancreatic beta cells. Glucokinase has got lower affinity and higher Km. Okay, now tell me what happens in liver. So in liver, in the presence of insulin, Okay, this insulin induces glucokinase. Insulin induces glucokinase. So in the presence of insulin, glucokinase in the liver will be stimulated. Only following that, in liver, glucose will be converted to glucose 6 phosphate. That reduces the intracellular glucose concentration of hepatocytes, and that will allow glucose uptake to happen into hepatocyte. Do you understand this? Though there is no GLUT4 transporter in liver, insulin stimulates glucose uptake into liver also, because insulin induces glucokinase and that causes glucose uptake into liver. Okay, so tell me what is the right answer here? Which of the following tissues are independent of insulin for glucose uptake? You know, liver is dependent on insulin, muscle is dependent on insulin, adipose tissue is dependent on insulin. So which is independent of insulin? Placenta is independent of insulin. Okay, answer is choice D. So quickly answer, I know all of you know the answer. Hooch tragedy. Hooch is a spurious alcohol, right? Hooch is a spurious alcohol. Whenever there is spurious alcohol synthesis, methanol gets distilled. During distillation of ethanol, by mistake, methanol gets distilled along with that. And following consumption of just 10 ml of methanol, they present with optic neuritis. They present with metabolic acidosis, right? So what is Hooch tragedy? It is because of methanol consumption. Okay. Which of the following is true? To break AT bonds, high temperature is required. To break GC bonds, low TM is required. To break AT bonds, low TM is required. AT and GC bonds need the same TM. First tell me what is TM. Have you understood the answer? Very good, Titir. Yes. Answer is to break AT bonds, you need lower TM or lower melting temperature. So what is TM, all of you? TM or melting temperature is the temperature at which 50% of the given double-stranded DNA undergoes unwinding. Do you all understand this? TM or melting temperature is the temperature at which 50% of the given double-stranded DNA undergoes unwinding. And how do you bring about unwinding? By breaking hydrogen bonds. And these hydrogen bonds get broken at higher temperatures. Now this TM is directly proportional to GC content. Because more the GC content, more will be the number of hydrogen bonds. I hope you all know that there are 3 hydrogen bonds between G and C and 2 hydrogen bonds between A and T. So if a segment of double-stranded DNA has got more GC content, then there will be more number of hydrogen bonds. So you will need a higher temperature to break it. So TM or melting temperature is directly proportional to GC content. It is inversely proportional to AT content. Okay, so what is the right answer? The right answer is to break AT bonds, lower TM is required. So now, many of you were asking me to discuss molecular biology MCQs because in INI-CET May 2022, just a year back, there were just 7 MCQs in biochemistry. Okay, previously it was 17 MCQs and the very next INI-CET there were just 7 MCQs. Out of these 7 MCQs, 5 were from molecular biology. And this was the exam, this was the INI-CET which was considered as the most difficult INI-CET ever. Primarily because there were many molecular biology based MCQs. Okay, so I'm going to help you answer those MCQs today. Sanger used dideoxynucleotide triphosphates for sequencing instead of deoxynucleotide triphosphates because of what? What is the right answer? We call it as, Sanger's termination method. We call it as Sanger's termination method. So tell me who sequenced protein first? Who sequenced protein first? Sanger. Who sequenced DNA first? Sanger. Okay. And what is the protein Sanger sequenced first? Sanger sequenced insulin first. Okay. And as far as Sanger's DNA sequencing is concerned, the technique is called as Sanger's termination method or Sanger's dideoxynucleotide triphosphate method. The principle is very simple. The principle is the moment dideoxynucleotide triphosphate gets inserted, there can be no further elongation by DNA polymerase. So what is the right answer here? The right answer here is insertion of dideoxynucleotide triphosphate will result in prevention of elongation of the chain by DNA polymerase. Okay, so let me tell you what it means now. Please watch this for the next 5 minutes with patience. If you watch this for the next 5 minutes with patience, you will understand exactly about Sanger sequencing. So what is the method called as? The method is called as dideoxynucleotide triphosphate method. It is also called as a termination method. And the principle that is used is whenever dideoxynucleotide gets inserted, there can be no further elongation by DNA polymerases. Why do you think there can be no elongation? What is the difference between deoxynucleotides and dideoxynucleotides? Deoxynucleotides means there is a deoxygenation which has happened only in the 2' carbon atom. Have a look at this. This is deoxynucleotide. Why am I calling it as deoxynucleotide? Here this is the 1' carbon atom, 2' carbon atom. In 2' carbon atom, do you see that there is no OH, just a H? Which means what has happened here? Deoxygenation has happened in the 2' carbon atom. So deoxynucleotide is nothing but 2' deoxynucleotide triphosphate. In which case, the 3' hydroxyl group will be involved in 3' 5' phosphodiester linkage formation. It's an uncoupler. No, it's not an uncoupler. Listen to this please. Titar, you will understand it now. So if it's a deoxynucleotide triphosphate, there is deoxygenation which has happened only in the 2' carbon atom. So there is a hydroxyl group in the 3' carbon atom. This will get involved in 3' 5' phosphodiester linkage formation with the next nucleotide. Whereas what is dideoxynucleotide triphosphate? I can see that you're not able to see it. How do I show you this? Okay, no. So can you see it here? So this is dideoxynucleotide triphosphate. What has happened in dideoxynucleotide triphosphate? Here not only the 2' carbon atom, even the 3' carbon atom have lost their oxygen. So is there any 3' hydroxyl group which can form 3' 5' phosphodiester linkage formation with the next nucleotide? No. When you cannot form 3' 5' phosphodiester linkage, elongation cannot happen further. So do you all understand this now? Yeah. So that's about Sanger sequencing. So understand, this looks like a simple fact. Okay, whenever there is dideoxynucleotide insertion, there can be no further elongation. But how can this simple fact be used for sequencing? I'm going to show you this example. Okay, so watch this. Suppose this is the oligonucleotide that you are trying to sequence. Okay, so this oligonucleotide has got 10 nucleotides and the sequence is CTGA, CTT, CGA. You are trying to sequence it. What is step number 1 for any sequencing? Step number 1 for any sequencing is in electron transport chain, that is called as 2,4-dinitrophenol. Do you remember that, Titan? In electron transport chain, the uncoupler is 2,4-dinitrophenol. Okay, and here it is dideoxynucleotide triphosphates. Both are different. Got it? Okay, so what is step number 1 for sequencing? Step number 1 is you have to subject the oligonucleotide or the polynucleotide that you are trying to sequence to PCR. And you get multiple copies. Okay, after you get multiple copies of the fragment which we are trying to sequence, we add equal number of copies to 4 test tubes. Okay, and all these 4 test tubes will have all the requirements of PCR. What are the requirements of PCR? You know that PCR is an amplification process or PCR is a replication process. And for any replication, the first requirement will be a DNA polymerase. So what will you add in all the 4 tubes? In all the 4 tubes, I will add DNA polymerase. I will then add primer. You know DNA polymerases always need a primer to elongate the strand. I will add all 4 deoxynucleotide triphosphates. So all the 4 tubes will have all the 4 deoxynucleotide triphosphates. I will then add magnesium or manganese to act as a catalyst. I will add buffer to maintain the pH. So this way I've told you the requirements of PCR. Can you all repeat it along with me? I will tell you Sanger's reagent, Pankaj. Let's finish this and then I will tell you. Okay. So tell me, what are the requirements of polymerase chain reaction? Please answer this along with me. The requirements of polymerase chain reaction are: number 1, DNA polymerase. You add primers. You add all 4 deoxynucleotide triphosphates. You need magnesium or manganese to act as a catalyst and buffer. We have done all these to all the 4 tubes. But these 4 tubes will differ in which dideoxynucleotide has been added to all the 4 tubes. For example, in the tube which is green in color, I have added dideoxy-CTP. In the tube which is yellow color, I have added dideoxy-GTP alone. And then dideoxy-ATP, dideoxy-TTP. So these 4 tubes differ only with respect to which dideoxynucleotide has been added. Okay, and after I add all these requirements, I will subject it to PCR. Sequencing is nothing but polymerase chain reaction. Okay, so I'm going to subject all the 4 tubes to polymerase chain reaction. Okay, now what will happen in the tube which is in yellow color? In the tube which is in yellow color, I have added dideoxy-GTP. So the Taq polymerase that I have added in this tube will start elongating this strand. When it starts elongating this strand, against A it will attach T, against G it attaches C, and against C it has to attach G. When it has to attach G, it has got 2 possibilities, it has got 2 choices. It can attach either the deoxy-GTP that is added in all the tubes, or it can add the dideoxy-GTP which has been exclusively added in this tube. It is just a probability. So if this tube has got 100 copies of the fragment that we are trying to sequence, out of these 100 copies, to 50 copies it attaches deoxy-GTP, and to the remaining 50 copies it attaches dideoxy-GTP. If it attaches deoxy-GTP, the elongation will continue. But if by mistake it has attached dideoxy-GTP, it stops there. And if it stops there, it is going to give you a fragment of length 3 nucleotides. Do you understand this? If it stops there, it is going to give you a nucleotide of length what? Just 3 nucleotides. Can you see that here? Should we study Harper? Okay, I will tell you all about that after we finish this discussion, Alisha. So because it stops in the 3rd nucleotide, you will see in this tube something of length 3 nucleotides. Okay, now what will happen to the remaining 50 nucleotides wherein elongation has happened? So if elongation happens, it continues beyond this. Against T you attach A, against T you attach A. Against C, when you have to attach G, again there is a confusion. Do I attach deoxy-GTP or do I attach dideoxy-GTP? So out of the 50 copies, to 25 copies I will attach deoxy-GTP, in which case elongation will continue. To the remaining 25 copies, I have no other go but to attach dideoxy-GTP wherein termination happens. If termination happens here, that is going to give an oligonucleotide of length 6 nucleotides. Do you see that here? I am going to get an oligonucleotide of length 6 nucleotides. Now what will happen to the remaining 25? I will continue the elongation. When I continue the elongation, I will continue happily until I reach the 10th one. So against the 10th one, which is C, irrespective of whether I attach deoxy-GTP or dideoxy-GTP, it stops there. If it stops there, I am going to have an oligonucleotide of length 10 nucleotides, which means in the tube which has got yellow color, in the tube which has got yellow color, I will have 3 oligonucleotides. What are the 3 oligonucleotides? One will be of length 3 nucleotides, the other will be of length 6 nucleotides, the third will be of length 10 nucleotides. This I will see when I run it through electrophoresis. Do I see electrophoresis being run here? Yeah, the yellow one. So if I look at that lane, I know that the length of this is 10. I know that the length of this is 6. I know that the length of this is 3. So why am I getting something of length 3 nucleotides? Because in PCR, as the third nucleotide, I have attached dideoxy-GTP. Why will I attach dideoxy-GTP? Only when in the template, the third nucleotide is C. Okay, so that tells me that the third nucleotide is C. Okay, and why has it terminated with 6 nucleotides? It is because as the 6th nucleotide, I have attached GTP. Why will I attach GTP? Because the template strand has got C. So now I know that the 6th one is C. Then the length is 10th, which means the 10th one is also C. So have you understood this? Yeah. So just by looking at this one tube, I know 3 nucleotides. I know that the third one is C. I know that the 6th one is C. I know that the 10th one is C. So this way, if I integrate the reports of all the 4 tubes, I will know which nucleotide is placed where. So this is how Sanger sequenced it. Got it? Have you all understood this? So tell me, what is the take-home message for all of you? If not C, what? If not C, they're in third position. If it is not C, then in the tube which has got GTP, the elongation would have continued, right? Suppose you're taking the tube which is green in color, where you have attached dideoxy-CTP. The same oligonucleotide is getting synthesized. So across A, you will attach T happily. Across G, you have to attach C. So if you attach dideoxy-CTP, it gives you one of length 2 nucleotides. Do you understand this now? It will give you something of length 2 nucleotides. And to the remaining 50 copies, it continues, right? It continues until it comes across this G. So this G is the 8th nucleotide. Against this 8th nucleotide, it attaches C and it stops. So this will give me only 2 oligonucleotides, one of length 2, the other of length 8. So that tells me that the second one is G and the 8th one is G. Okay. So I'm going to summarize about Sanger sequencing now. Okay. Please listen to this. So what is a summary about Sanger's sequencing? Sanger's sequencing method is called as Sanger's dideoxynucleotide triphosphate method. It is otherwise called as Sanger's termination method. Why is it called as termination method? Because whenever dideoxynucleotide gets inserted, the chain elongation stops in PCR. After dideoxynucleotide gets inserted, DNA polymerase will not be able to continue the elongation. So this simple principle was used by Sanger to sequence it. And for those of you who asked what is Sanger's reagent? So Sanger was the first person to sequence protein also. And for sequencing protein, the reagent that he used was 1-fluoro-2,4-dinitrobenzene. Yeah, what is Sanger's reagent used for protein sequencing? It is 1-fluoro-2,4-dinitrobenzene. What is uncoupler, Titir? Uncoupler is 2,4-dinitrophenol. Do you understand the difference? Okay. So can we end the session here? Because it's almost 2 hours now. Okay, so I'm going to end the session now. I'll come again. I will meet you in the month of April with another series of INI-CET special sessions. I will concentrate more on molecular biology and molecular diagnostics. Okay. So best wishes. As I said, please don't be anxious. Please be mindful. Every minute matters, but the minute that you spend should be mindfully spent. Okay. So it's not about the number of facts that you learn. It's about how effectively I've learned that fact, how effectively I've learned that concept. Yeah, I will take another revision session. Okay, in the next month. Okay, so thank you. See you all.